let's begin. I like to begin always with why. So why do we need transport? So why transport systems? What's the need for a transport system? And this builds on the idea of the need for exchange in animals. Once you have specialized exchange systems, that's addressing an exchange requirement. Getting oxygen, getting enough oxygen at a sufficient rate into the body so that all the cells can carry out respiration, but then also allowing a route by which to remove CO2 effectively, efficiently from all the cells, so as not to let it build up too much. And so that kind of serves the metabolic requirement of the cells, but there's one problem. And I will illustrate that, for example, with a fish. Okay, so we have a fish. Okay, now let's say the fish's exchange system is at the gills. That's great. Okay, so oxygen kind of enters the system at the gills, and CO2 exits the fish at the gills. Now, the way I like to illustrate the need for transport systems is to say, well, it's great for the gills, for all the cells at the gills. That's great. You know, there's a high rate of diffusion at the gills. But how then do we supply the rest of the fish? Okay, and my argument would be that the exchange organs are great at kind of getting oxygen into the organism, so to speak, and removing CO2 from the organism. But how do we get that oxygen that's just come in to the rest of the body? And that is where the transport system comes in. Okay, so you need a transport, especially when the organism gets quite big. Now that the oxygen's in the body, we can't just let it diffuse to the rest of the body. That's not going to work, right? Because of the concerns over surface area to volume, diffusion distances, and concentration gradients. They're all insufficient. So you need a transport system. And so what we'd have is we'd have an exchange organ, and from the exchange organ it would be the transport system that would allow the oxygen that's come in from the exchange organ to get to the rest of the body and to get to the other cells. And it would be the exchange organ that, vice versa, or by a similar argument, would then take the CO2 that's being produced at all the cells and get it to the exchange organ so that the exchange organ can effectively remove it from the body. Okay? So that's why we need the transport system. The next thing we'll discuss is how they work. Okay, so how circulatory systems work. Okay, now, so circulatory systems have three main components. Okay, but we'll get to that in a bit. All right, so we've got like a series of tubes, and in the tubes we have a transport medium that is whatever needs to be transported is within the fluid. Okay, the tubes contain the fluid, and the heart pumps the fluid around. So this is essentially how a circulatory system works. You have stuff, you have different locations in the body, so you have cells in one place of the body, and you have cells in another place of the body. So for example, this could be the cells at the gills, okay, doing the job of the gills. And these could be the body cells, the rest of the body cells, okay? And what we want is kind of CO2 that's produced here, we want it to move there, and oxygen that's kind of coming in at the gills from the water at these cells, we want it to move to the rest of the body. So basically what happens is that the oxygen that comes in, I'll use some color coding here. Okay, so the gills are very good. We've discussed exchange, but at the gills, the oxygen comes into the blood or enters the blood. And at the body cells, they're carrying out lots of respiration, and so they're producing lots of CO2, and CO2 is entering the blood over there. Okay, and the job of the transport system is to move this fluid around. Okay, so we're going to have, so this fluid that contains the oxygen and CO2, that's called the transport medium. That's the transport medium. These blood vessels, or these vessels, they contain the transport medium, and we'll see why. Okay, so we got these, we'll call them blood vessels, or just tubes in general, that works too. Okay, and finally up here we've got this heart or pump, to be general. Okay, and that's putting pressure on the transport medium, and it's moving the transport medium. But in doing so, what that means is we don't have to wait then for CO2 to diffuse to the gills. We don't have to wait for oxygen to diffuse to the body cells because the fluid is moving already in that direction. So it speeds up the transport. Okay, so the oxygen will then move to where we want it to go, and the CO2 is then moved where it needs to go. Okay, and that's the purpose of the circulatory system. It's basically giving diffusion a helping hand. Okay, because diffusion for a large multicellular, I mean that's an important idea as well, that for an organism that's too large for diffusion to effectively supply all the cells and at the same time remove all the waste from the cell. For organisms that are large, multicellular, very metabolically active, for example, they might have muscles, the nervous system, they're moving around. For organisms like that, not only do we need the exchange organs specialized to increase that rate of diffusion, but we also need the substances exchanged to be effectively transported. And that is the purpose of the circulatory system, and generally this is how they work. Okay, so what we're going to do is we're just going to go over that and make that apparent, and then we'll kind of, the idea here is that we look at those general principles first, and then we see how in different organisms with different types of circulatory system, how these general principles are implemented. Okay, so we have our transport medium. So the transport medium is important. Now, for example, like this, for many organisms, this is blood, but it's not always the case. But you have a transport medium, and that's primarily it's water. Okay, and it needs to do a certain number of things. It needs to be good at. It needs to be a good solvent. It needs to be a good solvent. It needs to dissolve biologically important molecules very well so that they can be transported. Okay, so water does that job. It dissolves things like glucose, it dissolves things like amino acid, it dissolves things like oxygen and CO2. And that's, for a simplistic discussion, that's all it needs to do, right? So it's a good solvent. There are, thermal properties that it has as well, but they don't really relate to transport, so I'm not going to talk about that too much. But that's the job of the transport medium. It must dissolve the substances that need to be transported. Okay. Next, we have the tubes. Actually, we'll come to the tubes next, but we'll discuss the pump. Okay. Now, the job of the pump is to contract. So it's muscular, and it contracts, and it rapidly reduces volume of its compartment, increasing pressure, okay? It increases pressure on the transport medium. It increases the pressure on the transport medium. And what this does is, because the transport medium is then at a high pressure, so we're going to have high pressure here, where the pump is, and relative to that, everywhere else is going to have a lower pressure. So for example, we'd have low pressure down here. So if we've got high pressure here and low pressure down here, then the fluid is going to move down the pressure gradient, okay? And so we get movement, okay? So the pump contracts, it increases pressure on the transport medium, getting it to move by what we call mass flow, okay? Mass flow. This is important because this is different to diffusion, all right? Though the idea of gradient still applies, but chemicals move from a high concentration to a low concentration. That is diffusion, right? But when you move from a high pressure to a low pressure, that is mass flow, okay? And I hope we can see, like that's much more energetically demanding, all right? Because we need all that energy to get the muscles to contract and so on. Okay, finally, we need the blood vessels. So these are our three components: the transport medium, the pump, finally the blood vessels. What's their job? Well, blood vessels, they contain, or they maintain, their main job is to maintain the pressure, on the transport medium. If the heart contracts, but the blood just squirts out everywhere, then we've lost the pressure. Then it's not going to really go very far. But if the heart contracts and the vessels contain that blood or the transport medium and keep the pressure on it, then the fluid is going to move over a longer distance. Okay? So the blood vessels have an important job then, because then as long as they can maintain the pressure on the fluid, it's going to keep moving, and that allows organisms to get larger but still have effective transport systems. Okay? So the blood vessels have an important job of maintaining the pressure on the transport medium. As long as they do that, the transport medium can keep moving, right? And so you can be as big as a whale and you can still get the blood moving from the lungs to the fins, okay? Or over great distances. So that's the other job. That's one job. The other job is, at some point, they have to allow diffusion. Okay, they have to allow diffusion. So when the blood does get to a point where the oxygen in it needs to be delivered or unloaded, for example, those blood vessels do need to allow the diffusion to occur. So they've got these kind of two functions to achieve, and we'll see how they do that. Okay, so that's the key aspects of transport systems. Now what we'll do is look at how they've been implemented in different ways. The next thing that we're going to look at is types of circulatory system, and just compare them and consider the reason why there's been this gradual evolution of the transport system. Okay, so as we've said, the key elements of a circulatory system, of a transport system, are the transport medium, the pump, and the blood vessels, and they are always present. But let's look at the different types of system that we're looking at. So the first one, usually in smaller organisms, but still relatively quite complex, still multicellular, so they do need a transport system. But insects, for example, have, and so we're talking about here, an open circulatory system. An open circulatory system, and it's usually found in organisms such as insects. Okay, so in this case we've still got the heart. It's still putting pressure on the transport medium. We've still got a transport medium, this fluid, okay, the hemocoel that moves around, bathes the cell, okay, and we've got the vessels, but the vessels are not closed, they're open-ended, okay. So what happens, again, the cells, for example, let's say that these cells are near a tracheole, okay, so these cells are near, there's a nearby tracheole here, okay, and so there's a high availability of oxygen over here, whereas these cells happen to be not maybe very, very close, relatively as close to a tracheole, and so maybe there's a greater concentration of CO2 here. The purpose of this system is to keep that fluid moving around the organism so that the CO2 can be moved perhaps closer to a place where it can enter the tracheole and then diffuse out, and the oxygen that's in the fluid near the tracheole, that fluid with a higher oxygen concentration can be moved around the body until it gets to places where there's a lower concentration of the oxygen, and so it can diffuse into the cells. So the purpose of this system is just to keep the fluid moving so that there's always a concentration gradient between the concentrations of gases in the fluid and in the cells. Okay, but the way that system works is how we have discussed. So it maintains concentration gradients. So I think it's helpful to think of it as what are the advantages of the system and what are the disadvantages. So in this case, yes, we're maintaining, it does maintain the concentration gradients, so it allows the diffusion to be maintained. The problem is, because those vessels are open-ended, there's only so much of a pressure that you can build up on that fluid. And because there's low pressure in those blood vessels, the distance that we can move the fluid is limited. This means organisms that have this kind of open-ended system can only be so big, because the pressure in these tubes is going to be lower. That means that the distance that we can effectively transport things is lower, and that eventually that's going to limit the size of the organism. So that's good for relatively small multicellular organisms like insects, but as we get to more complex, larger, metabolically active organisms like fish, for example, that will be insufficient. Okay, so what's the adaptation that we can make then to try and solve that problem? Well, next we go to a closed circulatory system, but it is a single circulatory system. There's one loop of the circuit for every beat or contraction of the heart. Okay, so it's closed and it's single circulatory system. So the advantage, it's great now because now we have a closed blood vessel system, and so the pressure that we can get on the fluid is higher, and so we can move fluid greater distances, and so the organisms can be larger. Okay, so larger organisms, yeah, they can be more metabolically active, and that's great, but there is still a disadvantage, and the disadvantage here is the pressure is limited. The pressure, we've got it higher than the open circulatory system, but the pressure is still limited because as the heart contracts and the blood moves to the gills. The exchange happens at the gills, the blood gets oxygenated, and then it goes to the rest of the body. But we can't indefinitely increase this pressure. If the pressure to the gills gets too high, so say if we want say if we want a larger organism, then we'd have to increase the pressure, right? So, but if we increase the pressure too high, it will damage the gills, right? So the pressure that the rest of the body gets is going to be limited by how much we can raise the pressure before we start to damage the gills. Okay, so the pressure that the body, the systemic pressure, is limited because we can't increase the pressure too high or it might damage the gills. Okay, so it's great, we've got it better than the open circulatory system, but the closed and single circulatory system, while maintaining a higher pressure, we're still limited by how high we can actually go. Okay, and so like organisms with this kind of circulatory system, they might be limited in terms of how big they can get, they might be limited with how active they can be. And so finally we get to the last system. This is the one that mammals have. So let's just give the example here. This is, for example, fish. Okay, but finally we move to mammals. And they have a double circulatory system, and some other organisms also have a double circulatory system, where there's, for every contraction of the heart, the blood goes through 2 circuits of blood vessels, okay? Or there's 2 circuits present. Now, in a double circulatory system then, what can be done is, again, we've got all the advantages of the closed system. We've got high pressure on the blood, so we can move it greater distances. We can maintain higher concentration gradients. But what we can do now is we can maintain a low pressure on the circuit that goes to the lungs, so we don't damage the exchange organs, right? So when on the circuit that goes to the lungs and back, we can have a low pressure or a relatively lower pressure. Whereas the circuit that goes to the rest of the body, because it's separate, it doesn't have to go to the lungs first before it goes everywhere else, it goes to the lungs and back to the heart. When the blood goes from the heart to the rest of the body, we can pump that under a much higher pressure without having to worry about any damage that might be caused to the lungs, because on this circuit the blood is not going past the lungs for exchange. Okay, and so we have that advantage. And so with the double circulatory system, we can have a higher systemic pressure, right? So we can have a higher pressure going to the rest of the body, and so organisms can be bigger, they can be more active, because there's a more effective supply of oxygen and removal of carbon dioxide. But at the same time, we can have a much lower pressure going to the lungs, and without damage to exchange surface. Without damage to the exchange surface. Okay, so those are our 3. And I guess the next thing that we do now is we focus on the double circulatory system in mammals, looking at the structures that are involved, and then we'll start looking at the functions. Okay, in this next part we're going to spend some time talking about the structures that relate to the mammalian circulatory system. Okay, and just to refer back to this briefly, just to confirm that in this system here, the vessels that are taking blood away from the heart are known as arteries. So that would be an artery. Okay, and this one would be an artery. Yep. And those blood vessels bringing blood back to the heart would be known as veins. So that would be a vein there. This one would be a vein there. Okay, and then appreciating that when an artery gets to or reaches a particular organ or particular part of the body, it will then branch out into capillaries, where we want exchange to happen. Okay, so the function of arteries and veins is transport, it is mass flow, whereas the function of capillary networks, that's exchange and it is diffusion. Okay, so there, when an artery transports the blood to the muscle, for example, it's going to branch out into capillaries where the oxygen can then come out of the capillaries and CO2 can enter the capillaries, and then those capillaries will converge back again into a vein whose main job is to then take that CO2 or transport the blood with that CO2 back to the heart. Okay, so when we look at the structures of the different blood vessels, it's important to take into consideration what their job is. So an artery, first, we can notice like structural differences. An artery has a narrower lumen, so we have an artery here, we have a vein here, and we have a capillary there. Okay, now the artery's got different structures because, yes, its main function is transport. So I'm just going to write it down here. Its main job is transport, get blood from one place to another place. But what's its particular challenges? Well, arteries are taking blood away from a heart that has just contracted, right? So it has to deal with high pressure. Okay, so it has to maintain the pressure, it has to withstand the pressure, two things. So it has to deal with high pressure, and so because of that, it's got a thick layer of collagen. It's got a thick layer, so it's got collagen because collagen is got a high tensile strength. Okay, it allows that blood vessel to withstand the pressure. Okay, it's got lots of elastin. It's got a thick layer of elastin so that it can expand and recoil to maintain that pressure on the blood. So the collagen gives it the strength to not burst under the pressure. The elastin gives it the ability to stretch a little bit and then recoil to maintain that pressure on the blood. Okay, because the longer we maintain the pressure on the blood, we keep the pressure on the blood, the further the blood can go. Okay, and finally it's got an endothelium in there. It's got a narrow lumen. Okay, narrow lumen. Again, this helps to keep that blood under high pressure. Okay, so then we go on to the vein. Now the vein, as you can see, it's got collagen. It's got, oh, I forgot to mention the smooth muscle here. So this smooth muscle also here. Okay, but in comparison, the vein has got much thinner layers of collagen. So it's got collagen, but much less. Less collagen, less elastin, wider lumen, and I hope you've noticed my version of what appears to be valves. So it has got valves. Now why? Now by the time the blood gets to the veins, that blood has lost a lot of its hydrostatic pressure, and the blood is now at a low pressure, but the blood still needs to be transported back to the heart. And so the vein has got valves to ensure that if the vein does get squashed, then the blood, because of the valves, the blood doesn't go in the wrong direction or backflow. The valves ensure that the blood gets forced in the right direction. The other thing is that its walls are much thinner because it's got less collagen, less elastin, and it's got a wider lumen, so collectively it's more squishy. Now what that allows is that as a person breathes in and out, or as a person moves, the action of the skeletal muscle around a vein squashes it, and that allows a little bit of additional pressure on the fluid to help that fluid move. But because that pressure is not coming from the heart, the valves help to keep that fluid moving in the right direction. So the combined action of movement of the body and the skeletal muscle squashing veins in particular places adds that a little bit of pressure to keep that blood moving, and the valves may ensure that that blood moves in the right direction towards the heart. So arteries and veins have the function of transport, but capillaries have the main function of diffusion. So the adaptations of capillaries are all to do with diffusion, not to do with high pressure, or not. They don't have valves to ensure the directionality of the flow. That's not the adaptations of the capillaries. The capillaries need to allow diffusion. So what do we see when we look at a capillary? We see a wall which is one cell thick. We see gaps between the endothelial cells so that fluid can move in and out. We see a very narrow lumen, barely big enough so that one blood cell could move through it, reducing the distance between the blood cell and the tissue fluid between which the diffusion is happening. So we're reducing diffusion distances. We've got a vast network, so an artery will branch out into many capillaries. That increases the surface area. Okay, so I'm just going to add that surface area network. Okay, so because a lot of branching out of the artery into many capillaries, we get a high surface area. We got cells which are one cell thick. We got the lumen which is very narrow, that reduces the diffusion distance. Okay, so the adaptations of capillaries are all to do with diffusion, and I hope that that's a concept that might tie it all together. Okay, so we've looked at blood vessels. Now we're going to look at blood, okay, and its components, and then we will eventually get to the heart. So the blood components, just looking at the structure of the blood. So what is in the blood? Key things: water. All right, what's its job? Water, plasma. Its job is to be a good solvent. Okay, next, big player, red blood cells or erythrocytes. Function: transport oxygen and CO2, which we'll get to in a bit. Platelets. Function: blood clotting. That's a bit of a defensive function there, platelets. Okay. What else do we have? We have phagocytes. And phagocytes have the function of being the primary defense. And finally, we'll have lymphocytes. Okay, and lymphocytes are part of the specific immune system. So those are our main specific immunity, and those are our main blood components and their functions, just to be aware of that. And now we will discuss the structure of the heart. All right, guys, so we move on to the structures of the heart. Now, I'm not going to spend too much time here because as we do the cardiac cycle, we're invariably going to talk a lot about the structure. So just a very, very quick one here. What I'll do is just number the structures. So let's actually, at the same, we could just talk cardiac cycle as we do this. We can discuss the route of blood flow. So blood, I always start here. Deoxygenated blood enters through the vena cava. Okay, so we have vena cava. Deoxygenated blood entering. So let's number that 1. So that is 1. Okay, and then it enters the right atrium. So that's structure 2. Structure 2 is going to be right atrium. Now, the next thing that's going to happen, the right atrium contracts, or the atria contract, and the deoxygenated blood moves through structure 3, which is the right atrioventricular valve, but often known maybe more specifically as the tricuspid valve. Tricuspid valve. Okay, then we are into the deoxygenated blood is then entering the structure 4, which is the right ventricle. Okay, ventricle's at the bottom. Okay, and then when the ventricles contract, then the deoxygenated blood is pushed through the pulmonary valve. So structure 5, how can I do this properly? I don't think I can actually. So right there, that valve is structure 5, and structure 5 is going to be the pulmonary valve. Pulmonary valve. Okay, and then the blood will go to the lungs. The exchange will happen with the air in the lungs, and the blood will come back oxygenated. So I'll get my red pen. Blood comes back oxygenated into the pulmonary vein. Oh, sorry. Forgot that one. So structure 6, so the blood is pushed, so the deoxygenated blood is pushed through the pulmonary valve into the pulmonary artery. So structure 6, pulmonary artery. 6, pulmonary artery, and that takes us to 6. Exchange happens at the lungs, blood comes back, oxygenated through, I guess we're on structure 7 now, the pulmonary vein, right? So structure 7, pulmonary vein. In it comes into the left atrium, right there. So structure 8, left atrium. Okay, then atria contract, blood pushed through the bicuspid valve. Hope I haven't got that wrong. I often do. The bicuspid valve, so that's structure 9. Okay, into the left ventricle, which will be structure 10. Left ventricle, thick muscular wall identifies it. Left ventricle then, and then when the ventricles contract, it's pushed through the aortic valve. Aortic valve into the aorta. So this right there, that will be my structure 11, which is aortic valve, and right up here, this will be structure 12, and structure 12 will be the aorta. Okay, so we've got the structures there getting busy, but this is not essentially for teaching. This is for recapping, this is for revision. So hopefully this is all familiar. The main purpose of this is to connect everything up with a reason. Okay, if it's just facts, it's going to be difficult to remember and revise, but if it's concept holding it all together, it's going to be much easier for you to remember, much easier hopefully for you to use. All right, so that's heart structures. Now let's move on to heart function, and we'll start with the cardiac cycle and move on to transport of oxygen, transport of carbon dioxide. Right guys, and we're back, and I am going to hurt my neck doing this bit. If only I was a little bit taller. Right, so we're going to talk about the cardiac cycle now, and I'm not going to draw out the heart all over again, but I am going to kind of note down the main features, what's going on, etc. With each of the stages. So diastole means relax. Okay, so I mean I like to begin with diastole. The heart is relaxed, it's not contracting, all compartments not contracting, which means there's a low pressure. Again, it's the reasoning behind all of this that kind of connects up the key facts. So the heart's relaxed, which means that there's a low pressure. If there's a lower pressure here than in the veins, then it's going to draw the blood in, right? Remember those pressure gradients that we talked about? High pressure fluid will move towards the area of low pressure. So if the heart is relaxing and it's low pressure, that means that the fluid is going to come in, the blood, the transport medium is going to be drawn into the heart. So blood enters the atrium. Blood enters right atrium through vena cava. Through vena cava. Okay, so there it comes in. Okay, the valves are also open at this time, so the ventricles are also not contracted, they are relaxed, and so these valves are open. So AV valves are open. AV valves are open. Just write that neatly. Atrioventricular valves are open on the right and the left side. Okay, so blood enters right atrium, but blood is also entering the left atrium's oxygenated blood. Okay, so blood is entering both the atria, okay, during diastole. Both the AV valves are open, so some blood is also filling up the ventricles. Okay, these valves are not closed. AV valves are open, some blood fills ventricles. So both the ventricles are filling with blood. So you often get like some exam questions where you're given a picture of the heart, the status of the heart at a particular point, and you have to be able to tell what's going on. Or you might be reading pressure changes on a graph. Okay, so come back to these things. Relaxed, so there's going to be low pressure. Some pressure might be increasing as it starts to fill with blood. Okay, and the valves are open, so yeah, you're going to get blood entering the atria and the ventricles at this point. Right, next we go on to atrial systole. So the next point in this is going to be the contraction of the atria, and we are going to eventually get to what controls all of these events. What makes the atria contract, what makes the ventricles contract, etc. We're going to get there. But during atrial systole, the atria contract. So that's the primary, both atria contract. That's the primary event that's going to trigger the next series of events. The atria contract, and atrial pressure higher than ventricular pressure. That's important. So the pressure, because the atria contract, the pressure here is higher than the pressure in the ventricles. And therefore, because of that, again, we come back to the pressure gradients. the fluid's going to move from the higher pressure to the lower pressure, and so the fluid moves from atria to ventricles. Okay? Actually, before that happens, the valves open. Okay, so because the pressure in the atria is higher than the ventricular pressure, the tricuspid and the bicuspid valves open. Okay, so the AV valves open, okay, and blood enters ventricles. Enters, blood enters the ventricles. Okay, that is atrial systole. Now, at some point, the blood is going to, the ventricles are now going to contract. So we enter ventricular systole, and now the ventricles are going to contract. So what are the key distinguishing features, and what's the series of events that happens during ventricular systole? So again, systole means contraction, so remember that. Okay, diastole, relax, systole, contract. Now we're in ventricular systole, which is the contraction of the ventricles. So ventricles contract. Okay, ventricles contract. So ventricles contract. So what? Well, if the ventricles contract, then now the ventricle pressure is higher than both the atria. So the ventricle pressure is higher than the atria pressure, okay? But it's also higher than the artery pressure. So the pulmonary artery and the aorta. Higher than atria and artery pressure. That's important, okay? So then why isn't the blood flowing back into the atria then, following the pressure gradient? Because valves, okay? Because of the structure of the valves, they allow flow from the atria to the ventricles, but not the other way around. So because this pressure is higher than that pressure, because the ventricle pressure is higher than the atria pressure, the atrioventricular valves close. The bicuspid and the tricuspid valves close. So AV valves close. Okay, so you have to think about this, and then you might have to identify what stage you're looking at based on the evidence here. So the AV valves close. However, these valves, the semilunar valves, the pulmonary valve, the aortic valve, because the pressure here is higher than there, it causes those valves to open. Now that's not something you have to work too hard to understand why. You can see from their structure that that's what would happen. Okay? So, semilunar valves open. Right? So semilunar valves open, blood moves from ventricles into arteries. Blood moves from ventricles to or into arteries, and out it goes. From this side, the deoxygenated blood will go to the lungs, and from the left side, the oxygenated blood will go to the rest of the body in the systemic circulation. Okay, one more thing though, in diastole, so when the relaxation happens again, during diastole, if we're saying that the heart pressure is low, why wasn't the blood moving from the arteries back into the ventricles? Because that could happen, right? Why? Because when the pressure is higher in these places, when the pressure is higher in the arteries just after a contraction, when the pressure is higher here than in the ventricles, the semilunar valves are closed. Okay, so just mention that here again. During diastole, the semilunar valves are closed, which prevents the blood from the arteries back flowing into the ventricles, even though the heart pressure is lower than those blood vessels. Okay, so that's the cardiac cycle, and I guess the next thing we should do is discuss why. Why did the atria contract first and then the ventricles? So that's what we'll do next. And we'll kind of discuss a little bit of ECG at the same time. Okay, so when we're talking about why did the atria contract before the ventricles, this is important. It's important that the atria do contract first before the ventricles, because you don't want all this stuff happening at the same time. It's going to F things up a little bit, right? Because what you want is the atria to contract first, essentially emptying into the ventricles. When the ventricles are fully loaded with the blood, then to contract and then push the blood out. If all of these things were happening at the same time, the ventricles might not be full with blood before they contract, and that would be a waste of the energy of the contraction, right? So the idea is that the atria contract first, and after a delay, we get the ventricles to contract. How do we achieve that? Okay, so let's have a look. I'm going to simplify the heart structure, okay, so that we're looking at something like this. All right, got the atria at the top, and so this is our right atrium, this is our left atrium, this is the right ventricle, and this is the left ventricle. Okay, now on the right atrium, in the wall of the right atrium, you know that there is that structure called the sinoatrial node. Okay, it's a myogenic little differentiated bit of the atria, which is specialized so that it kind of has its own rhythm of contraction set. Okay, so periodically it's sending out a wave of excitation through the muscle cells. Remember, these cells are interconnected, and when it does that, the wave of depolarization causes the muscle in that area to contract. And this wave of depolarization is being periodically just sent out at a baseline rhythm by the sinoatrial node. Okay, that's fine. So in event number one, so in diastole, in atrial systole, what causes the atria to contract is that initial wave of depolarization that goes out from the sinoatrial node. Okay, so that causes atrial systole. Okay. However, there is non-conductive tissue that separates the atria and the ventricles, which means when that wave of depolarization or excitation is spreading across the atria without any obstacles, when it starts to reach the ventricles, that non-conductive tissue prevents that electrical impulse from getting into the ventricles directly. So that is very important. This region of non-conductive tissue between the atria and the ventricles is important in preventing the ventricles from contracting at the same time as the atria. Okay, so that's the first thing. So we can call that event number one right there. Okay, so sinoatrial node, wave of depolarization or excitation, however you want to call it. Two is the non-conductive tissue, right? So the atria contract, but the non-conductive tissue prevents the ventricles from contracting because it just prevents the wave of depolarization from actually getting to the ventricles. Okay, however, the atrioventricular node, which is also around about on the right atrium, it does get hit by that initial wave of depolarization. It's not protected by the non-conductive tissue. So it gets excited, but instead of just passing it to the ventricles directly, it channels that excitation down through the Purkin fibers. So these are insulated fibers. Basically, they're protected or insulated from the ventricles, or the ventricles are insulated from the impulse moving down the Purkin fibers or bundle of His. So that, let's say that was 1. The 2 was a non-conductive tissue. The 3 is this idea that the impulse does activate or stimulate the atrioventricular node, but the stimulus then passes down the bundle of His, protected from the ventricles, okay, until the apex of the heart, okay? So 3 is atrioventricular node, 4 would be these bundle of His fibers which channel the excitation down to the bottom of the heart without letting it pass to the rest of the ventricles. But at the apex of the heart, okay, so when the excitation or wave of depolarization gets here to the apex of the heart, at that point it is allowed to spread to the ventricles. Now, by this time, at this point we've had a bit of a delay from the time that the atria contracted. There was a delay because it took some time for the excitation to get to the atrioventricular node. There was a further delay because the wave of depolarization had to pass along the bundle of His all the way down to the apex before it could then spread to the cardiac muscle cells of the ventricles. Okay, so once we get to the apex, then the ventricles contract. So that would be 6. Ventricles contract. Okay, so I mean the key thing there is, why do the ventricles contract after the atria? Okay, it's important, as we've seen, kind of maximizes the functioning of the heart in what it does, but how is right here. Right, this relates, so this electrical activity that's controlling the rate of contractions of the heart can be measured, and it kind of produces, if we can measure it, if we can detect it, it produces for us an ECG. So we're just going to spend a little bit of time looking at a normal ECG, sinus rhythm, and some variations that you can expect. All right, guys, we're getting there. Right, so we've discussed the electrical activity of the heart in relation to regulating one cardiac cycle, but then we could be asked to interpret ECG traces. Okay, now what we'll do first is just look at the components of a normal ECG trace, so like one beat of the heart, one contraction, one cardiac cycle. The electrical activity that's associated with that is as shown here. Okay, so you've got the P, then you've got the QRS, and then the T. Okay, now what you'd have to be able to do is recognize in the beginning, recognize what a normal trace looks like, but then what abnormalities can you notice? And the most frequent ones that you might have to identify are, I mean, there's two things that I would like you to look out for. The first is the rhythm. So the first is the rhythm. So this is, like here I've got a normal one, okay, so that's called a sinus rhythm. So if they're regularly spaced out and all the components of the normal ECG trace can be identified, not in my diagram, but you get what I'm trying to get across here, is that A, you'd be able to see all of the bits of the ECG trace, and they would have a particular set rhythm. Okay, so that's the normal one. But then you'd have to identify abnormalities. So what are the most frequent ones that come up? Now, this one you can see here that while all the components of the trace might be there, the beats or the contractions are more spaced out. So when you've got an ECG trace that has a much slower rate of contractions or beats, or much less frequent beats per minute than the normal sinus trace, then what you're looking at is bradycardia. Okay? Here, you've got all the components of the normal ECG trace are there, but they're much more frequent than the normal. So you do have to have an idea of what the normal appearance is and rate is. Okay? So, and that is tachycardia. Okay, and finally, what you have is two things. First, you can see the rhythm is gone here, so the spacing isn't regular anymore. Because in bradycardia and tachycardia, even though they had abnormal rates of contraction or frequencies of contraction, at least the spacing was regular. Whereas in this one, the spacing is irregular, and we might even lose a component of the normal trace. So some part of it might be there, it might not be there, or clearly identifiable. Maybe the T or the P. This is arrhythmia. Okay, and so these are the things that you want to look out for. The regularity of the ECG, and are there any parts of it that are missing? Okay. All right, let's move on now to look at the blood. There's the last few little bits here. The next thing that we're going to look at is in relation to blood, the formation of tissue fluid from blood, and finally the transport of oxygen and CO2 in the blood cell relating to hemoglobin. Okay, so let's begin then with that. So, I mean, this can be quite a long discussion, and it has been. But what we're going to do is we're just going to look at an artery, an arteriole, and a venule. And as we've said before, right, when the blood arrives at a tissue, the circulatory system, it branches out. The arteries branch out into these capillary networks, and that's essentially what we're looking at here. This artery has branched out into a capillary network, and they're going to then converge on the other side of the tissue. They're going to converge back into venules and then eventually into veins. So what happens here? So, and we're going to have some cells that will form the tissue around. Now I'm going to simplify things a little bit here, but we need to understand how the exchange process happens. Once the blood gets to the tissue, once it's been pumped out from the heart, that's not the end of the story. The story actually needs to be completed by what's actually going on at the other end when the blood gets to the tissues. Okay, so the blood's been pumped out of the left ventricle, it's moved a great distance in the artery, then it's got somewhere where it needs to be. What happens there? So the blood arrives oxygenated, okay, high pressure, let's call it hydrostatic pressure, high hydrostatic pressure. We'll use red for the hydrostatic. Okay, so the hydrostatic pressure is high, and remember, the capillaries, they don't have the layers of protection that arteries and veins do. Okay, so when you get to a capillary, stuff can leak out of the blood and stuff can come in to the blood from outside. So what happens? So you've got high hydrostatic pressure because the heart has just contracted, right? And the arteries have done their job and maintained that pressure all the way up until now. Okay, so we've got high hydrostatic pressure, so that's forcing blood out of the capillary the first chance it gets. Okay, so you got high hydrostatic pressure. However, there's a lot of stuff in the blood, right? Solutes, dissolved proteins, things like that, and that gives the blood a quite low water potential. Okay, so because of the stuff that's dissolved in blood, there is a force acting on the blood, acting on the water to come in. So while the hydrostatic pressure is forcing the blood or forcing fluid out, because of the water potential, there's actually a force that's kind of attracting the water inwards. We're going to call that oncotic pressure. But the point being is that at the venule side, or at the artery side, the oncotic pressure that's causing water to enter the blood vessel because of all the stuff that's dissolved in it, because of the low water potential, right? There's oncotic pressure. However, what we're saying is at the artery side, the hydrostatic pressure is greater than the oncotic pressure. So the force that's pushing water out of the blood vessel is greater than the force acting on the water to enter the blood vessel. And so the net movement of the blood, we'll indicate that in black, the net movement of the water is going to be out at the artery side. Okay, and great. And what this means is the oxygen, nutrients, glucose, amino acids, anything else, hormones, whatever, they will have a tendency at the artery side of this capillary network to, exit and enter the tissue fluid and allow access to the cells to get at all this stuff. So all this stuff then has a way to get to the cells and diffuses into the cells, and the cells can use it. But remember, the cells are also producing lots of waste stuff. They're producing things that we don't want to build up around the cells. Carbon dioxide, metabolic wastes, etc. The good thing is that by the time the blood gets to the venule side, the hydrostatic pressure, because of all the fluid that's left, the hydrostatic pressure is actually by now quite low. So yes, it's still there, right? But at this point, the oncotic pressure, which maybe I'll just draw it a little bit bigger. Okay, so at the beginning, the hydrostatic was greater than the oncotic, but now, not that the oncotic pressure has changed much, okay? It's still, in many ways, the same kind of size, but at this point, the oncotic pressure now is greater than the hydrostatic pressure. Or we could say that the hydrostatic pressure at this point has fallen below the oncotic pressure. Okay, so now at this point there's a tendency of things to move, or fluid to move net overall into the capillary just in time for it to be collected and taken back to the heart. Okay, so here at the venule side there is a tendency of things to move in to the capillaries, and so we get the process that we need, which is exchange. That's the key point here. Okay, so that's the key things to do with the tissue fluid. I mean, if you want an easy way to learn it, what I'd do is just say, right, what's happening at the arterial end? Okay, and what's happening at the venule end? Okay, so key points being hydrostatic pressure greater than oncotic pressure, therefore fluid moves out of capillaries. Okay, and all the nutrients and gases they contain, they move into the tissue fluid. But at the venule side, the oncotic is greater than hydrostatic, and so fluid enters, why do I keep writing entrees? Enters fluid, oh my god, enters capillaries. So overall, over the duration or over the distance of the network of capillaries, we're getting good two-way exchange. Okay, last thing then on the menu is the transport of oxygen and carbon dioxide involving hemoglobin and the red blood cells. All right guys, nearly there, and I will admit, after all this, I have run out of space, so we're going to try and get as much information in here, at least diagrammatically. Okay, so the next thing we're going to do is talk about how oxygen is transported in the blood, right? Because that's the whole point of this, or at least one of it. One of the key things that we're trying to do here is transport oxygen. Now, while water is a good solvent and it can dissolve oxygen, again, one of the evolutionary steps forward was to try and increase the capacity of the transport medium to carry as much oxygen as possible. And that's where red blood cells and their adaptations come in, right? Because hemoglobin increases the oxygen carrying capacity of the blood way beyond or much more than if it was just water. How do we display the oxygen carrying capacity of the blood? Okay, and that is this oxygen saturation curve, which what it essentially showing is reminding us that hemoglobin, remember, is this globular protein with a quaternary structure of 4 subunits. Each can bind a molecule of oxygen. Okay, but the more you increase the oxygen concentration, as it were, right, the more you increase the oxygen concentration, it doesn't go up in a straight line. It goes up in this sigmoidal curve. So when there's only a few oxygen molecules available, the affinity of the hemoglobin is low. But as oxygen molecules bind, it affects the shape of the other subunits and increases their affinity. So that the binding of oxygen causes the affinity of binding oxygen to increase. So it's like this kind of positive feedback effect. And the name that we give it is cooperative binding. So this exponential increase in the oxygen binding of hemoglobin is caused by every oxygen molecule that binds increasing further the affinity of the other subunits to bind oxygen. Why it levels off at the end is because we get to a point of saturation where there's, you've taken up all the oxygen binding sites, there's fewer around, and so there's not this indefinite increase in the oxygen binding. There is a cap on that, determined by the number of available binding sites. Okay, so that's the essentials of understanding that curve. There are some modifications to this, and it's important to understand this, that where, and even in the curve itself, when the blood gets to points in the body where there's not a lot of oxygen, what that means is we're going this way, right? So when the blood gets to, I don't know, like a heavily respiring tissue, what we're really doing is we're going this way on the axes, right? Because the oxygen concentration in those places is really low. So down we go backwards on the graph. And because we're in a place where there's not a lot of oxygen, the oxygen concentration is low, the affinity of the oxygen for the hemoglobin goes down accordingly. And so the hemoglobin has a greater tendency to release the bound oxygen so that it can go off, diffuse into the tissue fluid and into the cells that are requiring it. Okay? And similarly, when it goes to places like the lungs, where there is a higher concentration of oxygen, it's got a high affinity for binding oxygen. So when it goes to the lungs, it's better at binding oxygen. But when the blood, when the hemoglobin and the red blood cells get to places where there's low concentrations of oxygen, where tissues are using up lots and lots of oxygen, and they need it, well, in those places, hemoglobin has a greater tendency to let go its oxygens. So that makes a lot of sense, okay, and is an important adaptation of that molecule, okay, and red blood cells in general. Sticking on that theme, what helps further is the idea that I'm going to add a line here for what the curve looks like in a higher concentration of carbon dioxide. So not only does the lack or availability of oxygen affect the curve, but the availability or concentration of CO2 also affects the curve. And if you increase the CO2, the curve shifts to the right. So if you increase the CO2, actually, I should do that in blue. So if you increase the CO2 concentration, it shifts the curve to the right. Now, this means that we have reduced affinity for oxygen, okay? So this curve is in the presence of CO2. So plus CO2, we reduce the affinity for oxygen, which means, again, when the hemoglobin and the blood enters a region of the body where there's a high CO2 concentration, it's got a greater tendency to give up that oxygen. And that makes sense. Okay? Another situation, fetal hemoglobin. Now, fetal hemoglobin, the curve of fetal hemoglobin is approximately that, looks like that relative to adult hemoglobin. Why? Well, if moving in that direction indicates a reduced affinity for oxygen, shifting the curve in that direction indicates so this is fetal now. Fetal hemoglobin has a higher affinity for oxygen. Again, that makes sense because you have the mother circulation and you have the fetal circulation. Now, the fetal circulation has to have a higher affinity if the oxygen is to shift from the mother's blood over to the circulation in the fetus. So in order for the fetus to be able to grab the oxygen, it has to have a higher affinity for the oxygen, and therefore it makes sense that the curve is shifted to the left. Okay? Again, spend some time with that, make sure it makes sense to you. Okay, finally, just going to talk about the other important job that the blood as the transport medium does, which is the transport of CO2, the transport of carbon dioxide. Because this is kind of like the journey there, right? The transport of oxygen is like the story as we go to the tissues, but as we go from the tissues back to the heart and then back to the exchange organ, we do need to talk about the other thing that the blood does, which is take the CO2 from the respiring tissues, get it out of there to the place where it can be removed from the body, which is the exchange organs. So when CO2 is picked up here, right, when it's picked up as the CO2 diffuses into the capillary at the venule side of the capillary network, what happens? So CO2, 5% of it is dissolved in the plasma of the blood, okay, as just dissolved CO2. But most of it diffuses into the red blood cell. in the red blood cell, a lot of it is going to, or at least a good proportion of it, up to about 20% of it, is going to bind directly to the side chains available in hemoglobin and just bind to it and be carried along in the red blood cell, just attached to, or bound to the hemoglobin. Okay, so that form is called carb-amino hemoglobin, right? The carbon dioxide is attaching to the available amino groups in the hemoglobin molecule. But the greater percentage of it, about 70%, is going to be converted into hydrogen carbonate by the enzyme carbonic anhydrase. I'll just jot it down here. Carbonic anhydrase. I'm sure you can't read that. Okay, carbonic anhydrase converts, the enzyme in the red blood cell converts some of the dissolved CO2 in the red blood cell, in the cytoplasm, into hydrogen carbonate ions. Now these will dissociate into protons. Because it's an acid, it's going to dissociate into protons and HCO3 or carbonate ions, okay, and which is negatively charged and the protons being positively charged. Now these will diffuse out into the plasma, the hydrogen carbonate ions or the carbonate ions, and in return the chloride ions move into the cell. It's just to kind of, because if the negatively charged ions just keeps going out, at some point we'll get a gradient of negative charge outside and positive charge inside, which might prevent more HCO3 from being able to exit, which will limit the capacity of the red blood cell to actually transport CO2, right? Which is bad. Because you want to be collecting up as much CO2 as possible to take to the exchange organs, the lungs, gills, etc. Right? So you don't want anything to be limiting that. So to maintain that electrical balance, chloride ions enter, which maintains this electrical balance, which means we can keep converting this carbonic acid into carbonate ions, and they can keep and so we can increase the capacity of the red blood cells to transport CO2. Okay, so this shift of chloride ions in means that we can keep chucking HCO3 minus out. Okay, right. What happens to the protons? Well, the protons, they actually attach to the hemoglobin, converting it or in a form that's called hemoglobinic acid. What's the significance of that? Well, the important thing that would happen as a consequence of this is going to change the tertiary structure a little bit, right? We know that the effect of pH on proteins is to alter the tertiary structure, and the attachment of these protons to the hemoglobin would change its tertiary structure in a way that affects its ability to bind oxygen. And that's where we come back to that curve that we discussed here. The effect of CO2 on hemoglobin is essentially explained here, right? So this reduced affinity that hemoglobin has for oxygen in the presence of CO2 is due to the protons produced, or as a result of the CO2 coming in to the red blood cell. And again, that fits in with the narrative that, you know, in the places where there's lots of CO2, we do want to get rid of the oxygen because these are the places that are actively or, using up oxygen in great quantities or at a high rate. Okay, so a lot of the CO2 then, just know the three forms that it's transported. 1, dissolves in the plasma as CO2. 2, binding to hemoglobin directly. 3, or hydrogen carbonate ions. And guys, that is transport in animals, central premise, structures, types of systems, types of kind of adaptations of that system, structures involved in the mammalian system, heart, blood vessels, blood, you know, cardiac cycle essentially being, you know, a deep dive into the function of the heart, right? And all, you know, we can look at the blood stuff, we can look at the transport of oxygen, transport of carbon dioxide, formation of tissue fluid as a kind of a deep dive into the function of the blood. And yeah, guys, thank you. I hope it's been useful. The transport in plants 1 should be coming up shortly.